converted and transported in blood. And then we're going to talk about how respiration is controlled by the nervous system. Now I know, the nervous system is primarily the focus of the other A and P class. So some of you have an advantage because you've already learned a lot about, the nervous system physiology and anatomy. But we have to talk about the nervous system here to talk about control of respiration, sort of the way we had to talk about the sympathetic nervous system when we were talking about control of the heartbeat. So let's see. I'll review where we were at the end of class on Tuesday. Carbon dioxide is produced by metabolically active cells. It's produced by pyruvate oxidation, by the pyruvate dehydrogenase complex in the mitochondrial matrix that produces acetyl-CoA, and then bringing up all kinds of wonderful memories from cell phys, right? Acetyl-CoA then feeds into the citric acid cycle, which produces, it produces NADH, FADH2, and some ATP. That's the energy conversion that is productive, but it also produces CO2. So for each molecule of glucose, right, there are 6 molecules of CO2 that are produced. Anyway, CO2 is produced in energy conversion in cells, especially in mitochondria. So the CO2 is a gas, but it is dissolved it's dissolved in the plasma. Well, it's dissolved in the mitochondrial matrix, and then it diffuses through the mitochondrial membranes, and is dissolved in the cell cytoplasm, and it diffuses through the cell membrane, and it is dissolved in the interstitial fluid surrounding cells, and then it diffuses through the endothelial cells that line a capillary, and then it is dissolved in blood plasma, right? It's dissolved gas, and it diffuses through the cell membrane into the red blood cell, into the erythrocyte, where it is dissolved in the cytoplasm of the red blood cells. So there is dissolved in the blood, CO2 is carried, some CO2 is carried as dissolved gas, both in plasma and in erythrocyte cytoplasm. But the majority of carbon dioxide is carried as bicarbonate. Most of the carbon that is carried by the blood is not carried as CO2, it's carried as bicarbonate ion, as follows. So CO2 can combine with water to form carbonic acid, and this is accelerated millionfold by carbonic anhydrase. Carbonic acid dissociates. There is the bicarbonate. The bicarbonate exists as a soluble solute inside the red blood cell cytosol, but it is also transported. Remember, I asked you, and you can redraw this on the handout for today. Draw a circle here, to indicate that this is not diffusion. So this arrow, all of these other arrows, this arrow through this membrane and this arrow through this membrane was just simple diffusion. This arrow is not diffusion. This arrow is antiport, secondary active transport of bicarbonate in exchange for chloride. Antiport. There is a huge number of these antiport proteins. It's one of the most highly expressed protein in any membrane of any cell. Red blood cells have just a huge number of these transporters. And so the bicarbonate that is produced inside the red blood cell is transported where it now exists as a solute in blood plasma to the tune of about 26 millimolar. Right? That's bicarbonate in plasma, at least in arterial blood. Now, in capillaries, we're not really talking about arterial blood, so it's even higher than that in capillary and venous blood. So then this blood now carries carbon dioxide in 3 major forms. It carries it as dissolved gas, either in the plasma or in the cytoplasm of red blood cells. It's carried as bicarbonate ion, either in the cytoplasm of red blood cells or in the plasma, a dissolved solute, and it's carried as a molecule that is bound to hemoglobin. Those are the inside red blood cells. Those are the three ways that carbon dioxide is carried by blood. And then, of course, the blood is transported by blood flow to the lung capillaries, where the process basically reverses. So now we're moving on into new material here. This is now no longer a repeat of Tuesday. Let's talk about what this is. So here's hemoglobin with carbon dioxide bound. What do these arrows represent? Dissociation, unbinding. So now we have CO2 as a dissolved gas inside the red blood cell, and of course we have CO2 as a dissolved gas in blood plasma, right, even from the start. Okay, this CO2, what is, so CO2 came as dissolved CO2, either as dissolved CO2 from the beginning, or now unbinding from hemoglobin. What is going on here? What is going on right here? That's the antiport, running in reverse. So I want you to draw a circle here and fill it in. It's not diffusion. It's the chloride bicarbonate exchanger running in the opposite direction. Bicarbonate is brought into the red blood cell, and chloride is transported out of the red blood cell. Exchange. And this bicarbonate combines with protons in the cytoplasm of erythrocytes. So what is this arrow? Well, I guess you'd say that's association. Right? This arrow right here has to be association of bicarbonate and protons. Association into carbonic acid. What is this? Carbonic anhydrase accelerating the reverse reaction. Remember from cell-phys that enzymes don't force a reaction one way or the other. They simply accelerate reactions. And now carbonic anhydrase is accelerating the separation of water and carbon dioxide as a dissolved gas. Question, Michael?
Is there a chloride gradient in both cases?
So there is typically not very much of a chloride gradient. The chloride, so this is a secondary active transport, but it's actually doing passive transport in both directions. This is bicarbonate moving down its concentration gradient in exchange for chloride, and this is bicarbonate moving down its concentration gradient in exchange for chloride. Why is this, so it should be clear why this is moving down its concentration gradient. Because the CO2 that was produced is diffusing, getting converted to bicarbonate, so there's lots of bicarbonate here because there's lots of CO2 here, and that drives this as a down the concentration gradient movement. Is that clear? Now, it may not be so clear why that concentration gradient is reversed here, but let me finish the story, and I think it will be. So this CO2 that is dissolved in the red blood cells and can diffuse passively, equilibrate with plasma, it's now diffusing from blood plasma through endothelial cells, through type 1 alveolar cells, into the airspace of alveoli, and getting ventilated out. So this is a concentration gradient of CO2 moving. Remember, this alveoli is being ventilated with air from the atmosphere that is very, very low in CO2. So this CO2, the amount of CO2 in the alveoli is low. It's being ventilated out. So this CO2 is moving down its concentration gradient. So this CO2 is moving down its concentration gradient. So this bicarbonate is moving down its concentration gradient. Some of this is diffusion, and some of this is transport by the antiport. You should know which is which. So that's the story of carbon dioxide transport and then ventilation by the lungs. Are there any questions about that? Okay. So then let's move on and talk about the control of respiration. So remember your friend, the Davenport diagram? We had I'm just taking an example, metabolic alkalosis. How could we get metabolic alkalosis? Kidney disease, excessive vomiting, something in the body that is getting rid of too much acid. And so the body is becoming too alkaline. Then there is respiratory compensation. That respiratory compensation is the control of respiration. Right? So when we talk about how is respiration controlled, we're talking about how we can compensate for a metabolic alkalosis or metabolic acidosis. That is to say, exercise would cause metabolic acidosis by itself, but we have a mechanism for compensating control of respiration that brings the pH back to normal. So now we're going to talk about those mechanisms. The mechanisms depend on the nervous system. And we have sensors in our body that detect CO2 and oxygen. So and they're called chemosensors or chemoreceptors. And you can talk about chemosensory cells or chemoreceptor cells. We can also talk about chemoreceptor molecules, but let's focus at the cellular level right now. So we're talking about cells that can detect and respond to changes in oxygen and CO2 in response to the body's use. So you exercise, you need more oxygen, you are producing more CO2, there is a respiratory response. We have two classes of chemosensory cells. One of them we call peripheral, and the other we call central. Central means it's in the brain, and peripheral means it's not in the brain. So let's talk about the peripheral chemosensors, or chemoreceptors. There are two primary locations of the peripheral chemoreceptor cells. One of them is very near the bifurcation of the carotid artery. So here's the common carotid artery. You should know the name of this tube right here. Brachiocephalic. Brachiocephalic splits into the right subclavian and the right common carotid. This should be ringing a little bit of a bell. The right common carotid then splits into the internal and external carotid arteries, and at that split, there is a structure called the carotid body, and in the carotid body are cells that are sort of tasting the blood. Not tasting, it's not taste, but they are sensing CO2 and oxygen. On both sides. The yellow things here are nerves. So those chemosensory cells are connected to nerve cells that carry action potential-based information from the cells to the brain. We also have chemosensory cells in the arch of the aorta. So right there, embedded in the endothelium of the aortic arch, are another set of chemosensory cells that are very similar to the ones that are in the carotid bodies. So those are the anatomical locations of the peripheral chemosensors. The central chemosensors are in the brain, and specifically they're in the hypothalamus. So some of you who have taken AMP1, this is a midline sagittal section of a brain, anterior, posterior, this is spinal cord, front, back, front, so this is left hemisphere, right hemisphere is out here somewhere. Frontal lobe, corpus callosum, I'm just, those of you, many of you have heard these words before. Corpus callosum, lateral ventricle, thalamus, hypothalamus, right there. That is the location of the central chemosensors. I won't test you on neuroanatomy for this course, but knowing the name of the location of the central chemosensors is inaccurate. Hypothalamus. So now, let's walk through a flow diagram of how these chemosensors control respiration. Your book does this as a flow diagram, and I guess I think it's pretty helpful. So let's start, in this case, increase in alveolar CO2. So what is the H word that would cause an increase in alveolar CO2? Hypoventilation. Hypoventilation. Ventilating the alveoli too little would allow CO2 to build up too much, and that would give rise to an increase in alveolar PCO2. P capital A CO2, if we wanted to use the abbreviation. Okay, let's walk through these. Let's be systematic about this. So what word describes why arterial, little a, P little a CO2 goes up in response to an increase in P capital A CO2? Diffusion. Thought I heard somebody whispering it. Alveolar and arterial PCO2 are almost always equal because of diffusion. Okay, so increase in arterial PCO2, hypoventilation. So there's an increase, since there's more PCO2 in blood, there's more PCO2 in extracellular fluid because of diffusion. So now in both of these, we have an increase in arterial acid, right? A decrease in pH, an increase in the concentration of protons in either blood, plasma, or extracellular fluid. Why? Why does an increase in arterial CO2 lead to an increase in arterial acid? Or does an increase in extracellular fluid, PCO2, cause an increase in extracellular acid? Why? Joe?
With the increase in CO2, the proton concentration goes up, which then...
Yeah, that's my why question. Why is that?
Does it have to do with the bicarbonate system?
It does have to do with the bicarbonate system.
So then, since the CO2 concentration is going up, then more H2CO3 is being produced, which then leads to HCO3 minus being produced, which then increases pH.
Right? It's just If you had said it's the carbon dioxide by carbonate equilibrium, I would have said, yeah, that's exactly right. Increase in CO2. I'm just asking you to reinforce this knowledge. Increase in CO2 will lead to an increase in proton concentration and decrease in pH. So carbon dioxide is in fact an acid. Okay, yeah, so I was just asking you to reinforce that bit of information. So we have an increase in proton concentration, a decrease in pH. Remember not to let pH and proton concentrations get you out of whack. Don't remember. Sometimes I will ask you about pH, and you have to remember, okay, an increase in pH is not an increase in proton concentration. It's just the opposite. An increase in pH is a decrease in proton concentration. Don't let the pH and the proton concentration thing confuse you. Make sure you're reading carefully. Are we talking about pH, or are we talking about concentration of H? Because they go in opposite directions. So I get confused when I look at this because I sort of automatically go to pH. Just that's the world I default to when I think about proton concentration. So I think about pH. But this is not an increase in pH. This is an increase in proton concentration. A decrease in pH. Okay, just word to the wise. Read the words carefully. Are we talking about pH, or are we talking about H bracket? Okay, so now we have an increase in H bracket, concentration of protons. And now you see here that we have in the peripheral chemosensors and in the central chemosensors. Now, where are these? Somebody tell me where these are? Carotid bodies and aortic arch. Good. And someone tell me where these are? In the hypothalamus. That's what I'm just getting you to say the words. And what does increase in firing mean in this context? Increase in the frequency of action potentials. And in the other AMP class, we talk a lot about, action potentials. We did some of it here when we talked about the cardiac action potential. You know that voltage-gated sodium channels are responsible for the upstroke of the cardiac action potential. Thinking about pacemaking, you know that if we increase sodium permeability, then we would increase the pace of the heart at the SA node, and that's an increase in action potential frequency. So the concepts that you learned, especially about the the SA node, are appropriate here for understanding why there is an increase in firing rate. There's an increase in the intrinsic depolarization of those cells to threshold, and it fires an action potential more frequently. So these cells increase action potential frequency, and they connect to the medulla. So let's go back. The medulla oblongata, integrates information from the hypothalamus and directly from those peripheral chemosensory neurons to control respiration. Respiratory control centers in the brain are in the medulla oblongata. So, there is control of the medulla oblongata. Basically, that's what these arrows are representing. Control of the medulla oblongata by the hypothalamus, and control of the medulla oblongata, at least parts of the medulla oblongata, by the peripheral chemosensors. And the medulla oblongata has neurons that control respiratory muscles. So the most important neuron in this arrow, respiratory muscle, the most important arrow between the medulla oblongata and contractions of respiratory muscle is the phrenic nerve. The phrenic nerve is what starts in the medulla, innervates the diaphragm. That's what the FHEs, diaphragmic. Right? Trying to make it make sense. Phrenic diaphragm. The phrenic nerve innervates the diaphragm, and the diaphragm is the single most important muscle as far as controlling respiration. There are other muscles that control this, the sternocleidomastoid and the scalenes and the external and internal intercostals. There are other neurons in here. But the single most important neuron here is the neurons from the medulla to the diaphragm, carried by the phrenic nerve. The phrenic nerve is now increasing the frequency of action potentials, which increases the frequency, well, not just the frequency, but the frequency of action potentials in the phrenic nerve increases both the strength and the frequency of contraction of the diaphragm and the other muscles. Increasing frequency will increase respiratory frequency, and increasing strength would increase the tidal volume. Increase in both frequency and tidal volume increases ventilation, alveolar ventilation. There's an equation that you should have in your head right now that's describing how you would calculate ventilation, V dot A, as a function of tidal volume, dead space, and frequency. I'll put it on the board if you want it, but you might already have it in your brain. You're going to use it on the problem set. So I'm not going to put it on the board because you're going to use it on the problem set. Increase in ventilation. Now we've just got a response. Increased ventilation. Increased ventilation will decrease alveolar PCO2 and increase, well, it will also increase alveolar PO2, but we're focusing on CO2 because that's where we started. And so this now normalizes alveolar CO2, and then all the fluids then respond because of the normalization of alveolar CO2, alveolar PCO2. So what's interesting is that we have chemosensors that are primarily responsible for controlling PCO2, but what they actually detect is pH, acid or base pH. Okay, let's do it. Oh, I forgot to bring the clickers, so you can use your fingers today. Peripheral and central chemoreceptor cells contain a channel that detects acid. And it's called ASIC, it's got a really great name, acid-sensing ion channels. They're activated by acid. These channels are selected for which ion? I'm asking you to apply what you know about the SA node to the chemoreceptor cells, knowing that these channels, remember what we got here is these channels lead to an increase in firing. Increased proton, I'm trying to mechanize, I'm trying to make a mechanism for why an increase in arterial proton concentration leads to an increase in the rate, frequency, of firing of these chemoreceptor cells. They have a channel that is responsible. What are these channels permeable to? They're selected for. Give me the finger. No, give me a finger. Oh. So many of you are getting this right, and some of you are not. They're permeable to sodium. An increase in sodium permeability would increase the rate at which these cells depolarize and bring them to threshold faster, and then so they would fire action potentials faster. So an increase in sodium permeability is responsible, just as an increase in IH is responsible, which are sodium channels. An increase in IH brings SA node to threshold faster. An increase in ASIC activation brings these cells to threshold faster. Different ways of sensing, sodium, different ways of controlling the membrane potential and bringing it to threshold. One depends on IH, a hyperpolarization activated sodium channel, and the other depends on ASIC, an acid activated sodium channel. But both of them bring the cells to threshold faster and cause a more frequent set of action potentials. If you chose carbon dioxide, that's wrong for a number of reasons. One is carbon dioxide is not an ion, so the question sort of gave it away. And carbon dioxide isn't going to change the membrane potential directly anyway. Carbon dioxide is what these cells are detecting, but what they're detecting is acid as a proxy for carbon dioxide. But carbon dioxide is not what these channels pass. Okay, is that clear? If these channels were selected for potassium, what would they do to the frequency of action potentials? They would decrease. Okay, so that was effective. So, yeah, let's take a break now. We'll take an early break today. Today's actually going to be a relatively short class, so let's take a 4 or 5 minute break, and we'll come back to this overview of sort of some of this flowchart is what we've done, and some of it is new. We'll do that after. Okay, questions about that? So here's a flowchart that covers all of the control of respiration. Some of this you know about. So here is CO2. This is, again, it's in hypoventilation, but hyperventilation, everything would be reversed. So we're just talking about hypoventilation, high arterial PCO2. High arterial or extracellular fluid concentration of protons, low pH, by the equation that I've erased, the equilibrium. We've talked about firing, and we've talked about the medulla. We've talked about neurons. So this one clarifies that the medulla has neurons that control the firing. Well, medulla neurons in the medulla that control the diaphragm. This is the phrenic nerve. So you can annotate your slide saying this arrow is the phrenic nerve. PHR. The diaphragm and intercostal muscles increase in frequency and strength of contraction. That increases ventilation. I just did what we did before the break. In addition to that, so that's major mechanisms for control, and it is both central and peripheral. In addition, there are peripheral chemoreceptors that detect arterial PO2. I think I said on Tuesday, the Nobel Prize in Physiology and Medicine was for cellular detection of PO2 and cellular responses, changes in gene transcription and responses to changes in PO2. And that's really important in how chemosensory cells detect PO2. There's a mechanism there, the molecular mechanisms for the detection of PO2 are complicated. There's a series of biochemical steps that lead to an increase in sodium channel permeability that leads to an increase in action potential frequency. But the mechanisms are complicated enough that, it's fun to teach, but it's not fun to learn. And so, so I'm not going to make you learn it. But you should know that there are chemosensory cells in the periphery. Where? Aortic arch and carotid bodies. These are sensory cells. So remember, those are clusters of cells. The aortic arch, there are clusters of cells. There are clusters of cells in the carotid body. Some of them are these kind of chemosensors, and some of them respond to oxygen. Right? Some of them are acid sensing, and some of them are oxygen sensing. and a decrease in oxygen leads to an increase in firing. But it's only in the periphery. The central chemosensors do not have oxygen detectors. They only have CO2 detectors, or acid detectors. So the peripheral chemoreceptor cells can detect decreases in arterial PO2 and increases in arterial proton concentration, which usually go hand in hand, but you know, there could be reasons, weird reasons for them to not go hand in hand. Questions about the flow chart, the control of respiration? What are the, let's have a conversation. What are the important parts of the brain? Give me one name. Hypothalamus? Another important part of the brain. Medulla? Another important nerve. Respiratory. Okay, so those are the three most important anatomical parts of the brain to know about for the control of respiration. Hypothalamus, medulla, respiratory. So let's talk about the medulla for a minute. So here is an image from another textbook of the medulla. And I'm not going to ask you to know any of this, but it's interesting because it reinforces some of the knowledge that some of you have, yeah, let me be optimistic, have from AMP1. So you might recall that, what do you suppose CN stands for in this? Cranial nerve 10. Where's cranial nerve 10? Doesn't even show. Oh, there it is. Cranial nerve 10 is the vagus nerve, for example. And some of you have learned the names and the functions of the cranial nerves. Many of the cranial nerves have their cell bodies in the medulla oblongata. And so let's talk about cells in the medulla oblongata that control inspiration. I'm not going to ask you to know this, but here are cells in the medulla oblongata that innervate cranial nerve 10 and cranial nerve 9 that control muscles in the larynx and pharynx. Here are nerves. This cranial nerve 5 is Ober's occupation, tropical trips. Trigeminal nerve. This is the trigeminal nerve. And it causes you to open your mouth and become a mouth breather if you need to inspire deeply, right? You may know that you sort of automatically start to breathe through your mouth when you need to. You can control it, but if you're not controlling it, you will typically open both your nostrils and your mouth when you need to breathe deeply during inspiration because of, let's say, exercise. Cranial nerve 7. Tropical trips aboard famous. Facial nerve. Facial nerve causes you to try to flare your nostrils, open your airway at your nostrils. Other muscles, cranial nerve 11, that's the accessory nerve. So other muscles, the accessory nerve innervates the shoulder, so the sternocleidomastoid and the scalenes is activated by cranial nerve 11. There's the phrenic nerve, other nerve. So the medulla, you have mechanisms that do what you might be aware of when you need to inspire deeply. You open your mouth, you open your larynx and pharynx, you open your nostrils, you activate the accessory muscles of the rib cage to drive inspiration. And then for expiration, mostly it's passive, right? But in forced expiration, there are muscles in the medulla that innervate the rectus abdominis, the abdominal obliques, and the internal intercostals to then force expiration if needed. And all of this is integrated. There's a really complicated set of neural networking to control the muscles of inspiration and expiration. Do you want the neurons that are controlling the diaphragm to fire at the same time as the neurons that are controlling the abdominal muscles? At exactly the same time? No, because firing the diaphragm is for inspiration, and firing the abdominals is for expiration. So there are ways to make sure that they are in alternating patterns. Those of you who took AMP1 know that we talked about alternating patterns in locomotion, left, right. Well, we have pattern generators in the medulla that control inspiration, expiration, inspiration, expiration. That's control of respiration. Questions?